Professor Yasser Abdel Hamid, and you can receive 1 or 2 questions from the floor. هو online. اتفضل سعادتك.
Thank you, Dr. Yasser, for your elegant lecture. My question as regards the renal Doppler resistive index. This means that I must know the normal resistive index of this patient, and then post-operative, the cardiothoracic surgery especially, to know the deviation from the normal, or just depend on a high value of resistive index. دكتور ياسر معنا؟
لا هي disconnected فمعذرة يعني.
دكتور ناجي. دكتور ناجي
نكمل اتفضل يا حضرتك القديم.
للاسف دكتور ياسر الظاهر ما قدرش يدخل اونلاين، هو كان مسجل المحاضره.
Thank you.
احنا thank you very much. احنا هننقل للمحاضره اللي بعد كده ان شاء الله. Professor Mohamed Salah. Okay, so I'd like to introduce Professor Mohamed Salah, Professor of Internal Medicine and Nephrology and Dean of the National Kidney Institute, Secretary General of the ESNT, and he's going to address potential novel biomarkers for early diagnosis and prognosis of renal allograft rejection.
Thank you, Mrs. Chairman. It's an honor to participate in the 40th annual conference of the ESNT, and we welcome all our guests from Egypt and outside the world. We hope that the conference will meet your standards in view of scientific agenda and organization. Our topic will be about the potential novel biomarkers for early diagnosis and treatment of renal graft. This is the National Institute where we celebrated the 25th anniversary, the silver jubilee of the Institute, and the 1,000 kidney transplant a week ago, or 2 weeks ago. The agenda will be introduction, definitions, and features of biomarkers of early and late graft dysfunction, biomarkers of post-transplant malignancy, of post-transplant infections, of post-transplant diabetes, biomarkers of post-transplant cardiovascular complications, and conclusion. So, clinical application of biomarkers is an integral component of transplant care. Clinicians and scientists are in search of a better biomarker than the current ones. We now use, example, the serological biomarkers. We use serum creatinine, donor-specific antigen or antibodies, etc., urine-derived urinalysis, urine protein, histologic biopsies, and so forth. The science behind recent biomarker discovery spans across multiple molecular biological disciplines, including transcriptomics, proteomics, and metabolomics. Innovative methodology and integration of basic and clinical approaches allowed researchers to unearth molecular phenomena preceding clinical disease. So what is a biomarker? It is, according to the WHO definition, is any substance, structure, or process that can be measured in the body or its products and influence or predict the influence of outcome or disease. And according to the National Institute of Health, biomarker definition is a characteristic that is objectively measured and evaluated as an indicator of normal biological process, pathogenic responses, or pharmacological responses to a therapeutic intervention. Proteomics are the systematic analysis of proteins for their identity, quantity, and function. Genomics is the study of the genome for estimating the risk for an individual to develop a disease. Transcriptomics is the study of expression patterns of all gene transcript, and metabolomics is the quantitative analysis of all metabolites of a specific biological sample. Biomarkers are subclassified into immunologic and non-immunologic. The immunologic is the representative of immune regulation and dysfunction, while the non-immunologic pertaining to delayed graft function cardiovascular events or mortality, infection, malignancy, post-transplant diabetes, graft, and patient survival. Novel biomarkers used to guide the diagnosis and management of transplant-related outcomes is a promising area for research. Chemokines are signaling proteins capable of inducing movement of certain cell types to areas of interest. Chemokines arise early in the immune cascade of rejection and thus can act as a biomarker to non-invasively identify deleterious immune events. Both urine and plasma chemokines have been studied extensively to detect immunologic dysfunctions as CXCL10 and CD30. Free microribonucleic acid are small, non-coding RNA segments integral to cellular function, while also present in homeostasis. In certain contexts, they signal perturbations at the molecular level, ergo are linked to disease. Free microRNA have been studied extensively in renal pathology, both in native and transplant kidneys. Given their regulatory roles and stability, both in vivo and in vitro, they exude potential as robust biomarkers. In their 2016 study of 160 patients, Matz et al. Showed that the expression levels of specific serum microRNAs, the 15B, 103A, and 106A, discriminated patients with stable graft function significantly from those with T-cell mediated rejection. Leukocyte subclasses, the predominance and activity of the different subclasses of leukocytes can indicate recipient immune status. Leukocyte populations thus can serve as biomarkers to detect and identify immune apparency preceding clinical disease. One such population is donor reactive memory B cells, or the MBCs. It is a subset of the B cell pool with emerging data supportive of a robust response to alloantigen post-transplant. In a 2018 study, MBCs were associated with rejection. Though the use of genomics in combination with histologic scoring, Yazdani et al. Were able to derive specific immune cell types and demonstrate that the presence of natural killer cells are predictive of antibody-mediated rejection. The leukocyte subclasses offer unique opportunities as biomarker in that they offer another vantage point into antigen-antibody dynamics that can occur independently of a preceding detectable donor-specific antibodies, highlights the role of less understood pathophysiology or pathophysiological mechanisms, natural killer cells, and their predictability of graft failure, and can potentially provide clinicians with an individualized recipient immune profile to guide management in terms of immune suppression. Gene expression profiles is an approach within the field of molecular biology whereby thousands of genes are analyzed simultaneously via messenger RNA to describe cellular function. Differential expression of genes, particularly those associated with immune cells and interleukins, are some of the earliest events leading to immune dysregulation and poor transplant outcomes. Consequently, these gene expression profiles can yield robust, viable biomarkers. Gene expression profiles biomarker is a predictive of subclinical acute rejection, persistent subclinical acute rejection, and acute rejection. A significant development in gene expression assays in kidney transplantation was the development of the kidney solid organ response test. And this is a 17 gene set created in 2014 that was found to detect acute rejection accurately. One of the most promising gene expression profiles is the TrueGraph molecular diagnostic test, which is a non-invasive test to surveil patients with a stable function that is now reimbursed by the Medicare out there in the US. The test was first validated in 2014, whereby Korean et al. Showed that the TrueGraph gene expression profile could distinguish patients with rejection from those with non-rejection dysfunction and excellent allograft function. Donor-derived cell-free deoxyribonucleic acid, the allograft transplantation can be considered genome transplantation with grafts having a unique allogenic signature. At baseline, cell-free deoxyribonucleic acid, DNA, is circulating at low levels. However, in the case of injury, including rejection, increased high levels of cell-free DNAs are shed into the bloodstream and are thus measurable as a biomarker. Given this recent quantification, donor-derived cell-free deoxyribonucleic acid is a nascent area of research. AlloSure is commercially available and reimbursed test by the Medicare. So what are the features of an ideal biomarker? What we are hoping to find? It should be a non-invasive and easily measured. It should be short turnaround time, that it does not take so much time to get a result. It should be an easy to interpret, reproducible and standardized, accuracy, sensitivity and specificity, good prognostic performance, poor proof of cause, and cost effective. So these are the ideal things we're looking for. Why do we need a biomarker to detect acute rejection? Well, alterations in serum creatinine are not specific for graft injury. A change in the level may indicate an intrinsic renal process such as acute rejection, graft infection, or a transient process such as hemodynamic effects of calcineurin inhibitors or pre-renal volume depletion. The process of acute rejection involves different stages with clinical signs of graft damage occurring late in the continuum after a stage of subclinical graft damage. In other words, serum creatinine may not change despite considerable injury to the kidney. So the performance of a biopsy may lead to, as we all know, complications. It requires hospitalization and it is expensive, it's invasive, other limitations. Example, sampling error, and a sample might not be of renal tissue, not of cortical tissue. The interpretation of this slide is operator dependent or observer dependent. It might differ from one operator or one pathologist to another. So the biomarkers here are paving the way for substantial advances, such as the possibility of accurately predicting risk of delayed graft function before graft is implanted, or of making a molecular diagnosis of subclinical rejection even before histological lesions develop, or of dissecting etiology of coronary artery disease, let's say. Identification of chronic allograft dysfunction, Identification of immunoquiescent or even tolerance patients to guide minimization of immunosuppressive therapy is another area of active research. Biomarkers measured in the immediate post-transplantation would be extremely useful to identify patients at risk of delayed graft function and prevent this common complication, for example, delaying start of CNI. Altogether, there is a need for alternative, less invasive but sensitive marker of diagnosis of acute graft rejection, identification and validation of biomarkers which correlate with and/or predict the presence of acute rejection and which could improve therapeutic decision making. By serial sampling, development of rejection might be anticipated before the actual development of tissue injury, and by being able to distinguish high risk from low risk, we can individualize the immunosuppressive medication for each. Some biomarkers have been analyzed in donors' biological fluids or in the graft, example, preservation fluid before kidney transplant, whereas most of them were studied in the recipient after kidney transplant for those who perform the cadaver transplantation and they use the preservation fluids. The potential biomarkers here, I picked those that we might use because the others are only for cadaveric, is the mitochondrial DNA, complement C5a, furosemide stress test, messenger RNA 182.5p and 21.3p, and other subclasses of messenger RNAs, and NGAL, we all know the NGAL, and choline, TL4 surface expression, amylase, and facin and vimentin. EVs is a general term which includes membrane structures of different size released by cells after fusion of endosomes with plasma membrane, exosomes shed from plasma membrane, microvesicles, or released during apoptosis, apoptotic bodies. EVs are then taken up by neighboring or distant target cells, paracrine or endocrine effect, and mediate a wide range of physiological and pathological processes, including renal disease. EVs also exert pleiotropic immunomodulatory roles in kidney transplantation. Plasma and urinary EVs investigated as possible biomarkers of delayed graft function, and they showed promising results. Other potentials for potential biomarkers for acute rejection: urine and peripheral blood transcriptomics and renal tissue transcriptomics. Complement-related biomarkers, they are, the complement system is deeply involved in antibody-mediated rejection and can therefore provide potential biomarkers related to this process. The C4D deposition, كلنا عارفينه, has been considered the gold standard indicating activation of classical pathway of complement. However, all complement pathways have been proved to be involved in antibody-mediated rejection, and now we have the term of C4D negative antibody-mediated rejection. So an increased complement activation in a pre-transplant biopsy suggests a predictive value of complement activation. Expression of these complement components at the time of diagnosis of antibody-mediated rejection was associated with higher serum creatinine and more severe morphological changes. As further evidence, C5 blockade prevents antibody-mediated rejection and stabilized long-term renal function. EVs shed by endothelial cell expressing C4D are increased in antibody-mediated rejection and correlate with its severity and response to treatment. Urinary and serum chemokines and other potential urinary biomarkers, and all are the DNA I mentioned before. I will skip for the sake of time. Allogenic circulating B cell and T cell assays, peripheral blood messenger RNAs, and immune cell biomarkers, non-HLA donor-specific antigen. Can I have 2 more minutes? أنا كنت فاكر 20 minutes الليكتشر عشان كده. The non-HLA DSA, donor HLA specific antibodies, were initially identified as a major cause of antibody-mediated rejection, and this type of DSA has been extensively studied. كلنا عارفينه. In more recent years, preformed and de novo HLA specific DSA targeting G protein coupled receptors expression also studied, and now angiotensin II receptor and endothelin type A receptor are in progress. Chronic allograft dysfunction. Chronic allograft dysfunction is the main cause of long-term graft loss. We're trying to find another predictive biomarker for it, and there are some set of genes related to fibrosis, urinary gene expression, renal tissue genes, plasmatic messenger RNAs are also used for prediction of renal allograft dysfunction, IFTA, and tubular atrophy. Monitor the tolerance. Also, I will skip for the time. Cardiovascular events and mortality. Infection, BK polyoma virus infection, COVID-19 now, also the SARS-CoV-2 with the biomarkers interleukin-6, we all know, malignancy and the malignant, the biomarkers for malignancy, post-transplant diabetes also, graft survival biomarkers, patient survival, there are some biomarkers for patient survival. Conclusion, further innovation and integration of multiple disciplines, omics, transcriptomics, metabolomics, proteomics, will lead to advanced biomarker discovery and implementation, which in turn will signature the will be of implementation in the diagnosis and prognosis for the grafts in the future. Thank you.
Thank you, Professor Mohamed, for your excellent presentation.
Thank you.
So it seems that we are in the era of biomarkers and too many biomarkers are evolving.
Yes.
If I could ask you, which biomarkers would you choose for your patients to predict rejection?
Well, I showed in the lecture
You showed many of them.
No, yeah, the table, I was stressing on this particular point you mentioned, Professor May, that some of these biomarkers are not applicable now because they are mainly derived, or towards the cadaver transplantation. But for our patients, NIGAL is a biomarker that can be used. The messenger RNA biomarkers also now used. The C4D, it is a biomarker and we are using actually. So these biomarkers that might be in the very near future. The TrueGraph test is already there in the US and it is now reimbursed by the Medicare and it's used and it showed great success in prediction of the acute rejection. And I think this test could be the most desperately needed test to bring in Egypt in the near future because it is approved by the FDA. It is already applicable. It's not that expensive, by the way. The expenses of the test, when you calculate the expenses of the biopsy and the biopsy process itself and the pathology interpretation, it is almost half the price. So it is an applicable one, and we can talk to our colleagues in clinical pathology and laboratory field to try to provide it in Egypt in the near future, inshallah.
Thank you very much. Any question? Any compelling question? No? Okay, thank you very much.
Thank
Professor
you.
Mohamed.